Hello, everyone, and welcome to this first session of our metagenomics training program. So what we'll do today is I'll talk briefly about some of the basic things that you have to know about metagenomics. For some of you, this will be a review. For some of you, many things might be new, but I'll also point you to some resources on our portal where you can find important information either on the basic terms that we will use throughout the program, as well as some practical exercises that you can get started with. So like I said, I will talk a little bit about some of the terminology, some of the methods that are used to generate the data, and I'll also talk a little bit about the types of projects that we will review or case studies where you can really see how metagenomics is used. Now, together with myself, Dr. Raghavendran Lakshminarayan is going to be mentoring the program, and in the next session, he will talk to you more about the technical details. But also, this is a program that is designed for you to succeed, and so we're hoping that you will update your profile, let us know about your background, your research interest, or your industry interest, and really allow us to connect with you. Our team will reach out to you if you have any questions, and we have a support group on Skype or on Slack where we can interact and address any questions that you might have. So the purpose of today's session is to discuss the microbiome, how we can use metagenomic sequence analysis to study the microbiome and understand how microorganisms interact in their natural environment, looking at the community composition and structure, thinking about species richness, and understanding the functional roles that some of these microbial communities can play in many areas related to human health and disease, as well as biotechnology and agriculture. So the program is really designed to introduce different types of case studies that illustrate the way the field of metagenomics has been evolving. What are some of the practical tools and skills that you have to be able to analyze this data independently? And also, what are some practical tips, tutorials, and maybe skills that you should have to be able to integrate this quantity of information from different databases, from the reads in the FASTQ files, and also the different metadata that you have about your samples to produce plots that help you interpret the data biologically. So you'll be able to, by the end of this program, process a data set, understand the composition, ask questions about the microbiome, and review literature that will help you find answers and make biological sense of the data that you have analyzed. The way the program is organized is it's really divided up into several different stages. The first one is logic, then we will talk about practice, and then we will kind of transition into a project stage. So why is logic important? Logic is important because we need to understand what kind of data are we going to be dealing with and what kinds of questions could potentially be asked of this data. Now, different types of data represent different types of biological phenomena. And so we can generate data that describe this biological phenomena, but depending on what kind of data we have about maybe the phenomena, maybe the number of samples, or the way that we plan to process and analyze the data will determine what kinds of questions we can ask. So moving a little bit more into the practical domain, we will talk about different types of analytical approaches. Now, the analytical approaches can be divided into two types. The first one is processing, or really preparing the data for analysis. And then the second one is more on how to work with the processed data and make sense of it. And that can include several different things. So one of them is kind of a standard process, which we call a pipeline, where we take the input files, we go through several different steps, and we get some predefined output. In many cases, that kind of an output requires further investigation. And that is driven both by statistical analysis, visualization, machine learning, and sometimes it's driven by a biological intuition. And so that's where we cannot cover everything that potentially could be done, but this will be more based on a case study. So in each case, we will see what kinds of questions are being asked, what methods are being used to process the data, how is that data then analyzed, visualized, and annotated, and then what kinds of interpretation or conclusions we can arrive, helping to study and find relationships between phenotype and what we find in this microbiome or metagenomic data. So this will be covered in several different sessions. Some of the sessions will be live, like today. Some of them will be pre-recorded. Today's session is live. You can ask us questions. You can comment. If you have any specific things that you expect from this program, we would love to know. But in many cases, we want to give you the flexibility of going through the topics that we have planned. And so that is why all of the sessions will be recorded, but also sometimes you will have access to a structured material on our OmicsLogic portal, which will include text, images, video, quizzes, exercises with coding or running pipelines. And that's why it's important to get familiar already with the portal, how it works, and how the different content present on that portal can be leveraged. And there you will find some basic coursework, as well as practical exercises in R and Python. We'll primarily talk about R because it's extensively used in metagenomic data analysis, and also because it is providing us with very nice looking figures that we can directly use in research publications. But the online portal really covers the basics, some practical, and gives you some case studies that are structured around publications where experts have been analyzing the data and coming to those conclusions. So it's a good way to understand not only how to technically approach a subject, but how have experts designed, sequenced the data, and made the interpretation of the results. Together with the portal, we will be using the T-Bioinfo platform. So the T-Bioinfo platform is a point-and-click interface that allows you to analyze big data without having to download the data to your computer, without having to learn how to write code in Bash, and really connect multiple complex analysis methods into a process, a pipeline, where you can get results that are interpretable in a quicker way. So to demonstrate how this is used in the context of metagenomics, I will show you a brief example, and then we will talk about how that can be used either to study metagenomics or to understand the relationship between phenotype and the specific types of data that we will be working with. So let me transition from my slides to the actual portal. Now, if you have already signed up on the OmicsLogic portal and you have access now to the program, your account information should be should allow you to log in to this portal, to this platform called the T-Bioinfo platform. So I'll pause here. Thank you, Ojasvi, for sharing the link. Please let me know, those that are participating and want to follow along, if you were able to get into this link. And also, if you hover over this profile, if you can see your email address here on top, so you know that you're logged in. And maybe you can share that in the chat. Yes, no, maybe something like that. So I will give you just a couple of minutes, and then we will go on from there. Okay, if you still have not created an account, then I recommend going first to the Learn portal. And on the Learn portal, you can easily create a free account. Unfortunately, if you're just creating your account today, you won't have access to the portal, to the platform right away. It will take a little bit of time for you to receive those credentials. So if you don't have the credentials, you can just observe. But for the next session, I highly recommend to try and log in. And if you're not able to log in or you have some issues with logging in, please please reach out to our support team at support@pine.bio. All right, so let's move on. And here you will see a a table called areas of analysis. You will see here many different types of analysis included: transcriptomic data, genomic data, epigenomic data, etc. And right here at the bottom, we have a dedicated section for metagenomic data analysis. So metagenomic data comes in several different formats, and that's why we have them divided into several different sections. We'll talk about some of them, and we'll use some of them quite extensively to analyze data. You will find that the selection of methods is driven in many cases by the type of data that you have. And so we'll talk a little bit about the types of metagenomic data in just a few minutes. But today I will show you a brief example of a specific type of data called amplicon 16S ribosomal RNA gene, where we amplify the 16S ribosomal RNA gene to understand the composition of a sample. And so this link right here will give us access to a demo. In the demo, we can build a pipeline without having to upload data and right away get results, so we don't have to wait for the processing time either. So if you do have access, you can click on there and you can follow along with me. If you don't, you can just observe. And again, you will have an exercise for next time where you can follow along. Now, every time that you click on a button, you get a description that explains what are we going to do and how. And so in this case, we're going to be using the data-to pipeline. Data-to pipeline was developed specifically because this kind of data is noisy. It has a lot of issues that can make finding signal difficult. And so because it is made to denoise the data, we have to go through a whole series of steps to first clean up the reads and prepare them for the downstream analysis. And so that's what we're going to do. First, we're going to check our quality of the reads. Then we're going to select only reads that are paired. After that, we are going to merge those pairs together. Then we will assign operational taxonomic units to those merged pairs of reads and remove chimeric reads that came from several different locations. So they kind of create this hybrid that cannot be annotated. Once we do this, we have to use a database, a reference that helps us assign each one of those sequences to a taxonomic level. And the taxonomic level basically has a database of sequences, and the closest sequence helps us align the OTUs with the taxonomy. After we have that information, we want to understand what types of sequences do we have. And to do that, we have to create a phylogenetic tree. The phylogenetic tree in this case can be created using this APE package in R, which stands for Analysis of Phylogenetics and Evolution. Once we have that phylogenetic tree, we can then start kind of analyzing and integrating all of that information to perform statistical analysis and study the composition of our sample. And that can be done with a package called phyloseq. Again, we will go through these packages during our sessions, and we can use them in R or we can use them on the platform. But what they do is they integrate the statistical analysis with plotting, and we will see how this plotting helps us understand what kind of data we have. And to the question in the chat, yes, this video is going to be available for everyone to see, but especially important for those that are participating in the full program. And then we're just going to produce some interactive visualization on the platform that will allow us to study the data set that we have. Okay, so for those of you that followed along, your pipeline connects all of these dots together. And what you have in the end are output files that you can download. And they typically are produced from every step that we selected. There will be something from those steps. But also we have here several plots that we can view interactively. So the first important plot is the taxa proportionate abundance bar plot. Again, you will have a whole session dedicated to how this plot is produced and what exactly does it represent. But here briefly, I would like to point your attention to several important things. First of all, when we talk about metagenomic data that represents the microbiome, we're studying a whole community together. And because we have different levels of depth for the different samples that we have, samples here are on the x-axis, we're expecting a lot of variation. And so we want to perform normalization by looking at proportionate abundance and not direct abundance from these samples. So once we have them, we can now see that specific taxa. Again, here we have the full taxonomic classification written out, but as you remember, bacteria is the kingdom, right? So it's not going to change. But then after this, you see that some of these taxonomic levels are going to start to change. And so as we look at our proportions here, we can see this green taxa, which is Bacteroidetes, vary between our samples. And so the obvious question now is, okay, what does that tell us about the samples themselves, right? This is a lot of detail. How do we kind of look at this detail from a high level to understand what kinds of differences do we have? And to do that, and we're going to, again, go through this in detail in one of our sessions, we use a statistical analysis called non-metric dimensionality scaling, which is a plot in 3D that we can look at, and we can see here each one of these dots represents a sample that we just saw. So here we have the samples on the x-axis, and on the y-axis, proportionate abundance. Here in our NMDS plot, we have 3 dimensions that are from the non-metric dimensionality scaling, and the dots represent in the individual sample. So you can see that the samples are of 2 types. Chow, which is a standard type of feed used for mice, and HFD, which stands for high-fat diet. And so as we look around, we can notice 2 major things. The first one is that we have an outlier. And the second one is that actually, if we probably remove the outlier, we would be able to see some pretty good separation between our samples, right? If we find some kind of a way to position this plot, we could find a line that could separate the dots. So of course, that is dependent on removing this outlier. And that is really the essence of this type of analysis that we will go through in the full program. This analysis is going to be iterative, which means that you will start somewhere, you will perform some standard tasks, and then you will start asking questions. What do I see? And what does it mean? And if I understand what it means, what are some next steps that I should take to make sure that I'm getting something that I understand, makes sense, but also is reproducible and could tell me something about the biology that I'm trying to study. So this is a very brief example. Again, we will go into much more detail throughout the program, but it illustrates the essence of the type of bioinformatics steps that you would have to go through and understand to be able to work with such microbiome data.